In order to explore some of the changes that might occur with sickle cell anemia, what we did was we used this bioinformatics platform, which is called Correlation Engine. This used to be known as NextBio. It was bought out by Illumina. Illumina does most of the sequencing on this planet, and they're very good at data. And they probably make most of their money with the sequencing, but what they wanted to do was provide a tool that would allow their potential customers to get more out of their data. So they bought this company, and what makes this platform special is that Illumina knows data. And what they've been doing, unlike a lot of the other platforms out there, is they're collecting public data. So right now they have over 20,000 different array studies, RNA-seq, basically just lots of omics studies that they're now collecting from the public sphere. They've downloaded in their database, and what this allows us to do is to search all these 20,000 different studies for sickle cell anemia studies. And we can do that, and you just basically enter terms here. We did this for sickle cell. Some of the pathways that came up. Let's see. Oop. So basically what we can do is we can put a term in here, sickle cell, and what I did was I wanted to see some of the differences in gene expression between blood cell types. And what I looked for was blood cells from the sickle cell patients versus controls. And given here are the 7 different gene lists from the different studies that I found that looked at this. Some of these are platelets, whole blood, and I even have let's see, endothelial cells. I think those are progenitor cells. Anyway, what we can do is we can use all these different groups. They're basically looking kind of at the same thing. What are the genes that changed in all of these lists? And now can we find genes in all of these lists that that are different in the majority of these? We're kind of a wisdom of crowd approach. So what we can do is we can hit this meta-analysis. Actually, we are on this. So these are the 7 studies. When we look at the genes that are upregulated or differentially expressed in those, it'll give them right here. So given here on our top list, this gene, insulin-like growth factor 2, binding protein 2, changed in 7 out of the 7 studies that we looked at. In 6 of those 7, based on this red color, it was overexpressed, and only in the last one where it was underexpressed. And basically we can do that so anything red would be overexpressed in the diseased or the sickle cell subjects compared to the control. And it agrees pretty well with all these different studies, which is nice. Some of these are interesting. But again, what we can do now, and what I did was take this list, and this is interesting in its own right. One thing I want to show you is basically now that we have this list, this meta-analysis, we can compare it to all their other studies and all of what else that they know. So what we can ask the software is, if I knock down a particular gene, will it result in a gene expression that's positively or negatively correlated to the changes we're seeing in the sickle cell individuals? So now that we have a list, what we can do is we can basically pick those genes that are expressed or differentially expressed in the majority of these studies. So 4 out of 7 studies, and basically I can filter this pretty easily. Not only that, but I can take the median fold change and basically use that as my direction for these genes. And as you can see, they're pretty consistent here. And that's what I did. And basically what you can do is you can take this. So what I did was I found those genes that were differentially expressed in the majority of these, at least 4 out of 7. I ended up with about 1,200 genes. What I can do now is save that list and re-upload it into this program and basically compare it to all the gene lists that they already have in their system. You can also get some preliminary bioinformatics results. So you can hit this Biogroup Results. And what this will show you is those biological functions, pathways, particular binding sites that might be overrepresented in the genes that we're looking at right now. Did I hit it? There it goes. And this is kind of where I got the first clue that there might be something in these sickle cell individuals from a gene expression state that might be preventing viral replication, or at least giving them some type of immunity to viral attack. If you look here, this is basically showing us the genes, or the genes that are associated with these particular processes, groups that are overrepresented, that we wouldn't expect this many genes associated, say, with viral reproductive process by random chance. Not only that, but you can see that most of these genes associated with viral reproductive is going down. We can see some of that here as we go down to, let's see, genes involved in influenza viral RNA transcription and replication. Those are also going down. So what I did, to go back to the gene results, I took these genes and basically took the ones that were the majority, and then I made my own list, which is here. And what I want to show you is, now that I have a bunch of genes that I'm very confident change in blood cells associated with sickle cell anemia, now I can analyze these for basically products. So basically I can put this list back into the system and ask it, say, what diseases are most like my gene list, based on not only the genes that I've put in, but the direction it goes. You can go to these correlated studies. That's obviously a really good place to look. Basically, it'll give you all those gene lists that it has in its database that resemble ours. And you can see that there's a lot of, not only is there peripheral blood, but there's things involved in influenza. So again, viral, which I find fascinating. I want to show you one more thing with the base space program, is that you can do this. What you can ask the program is, hey, I have this list of genes that are going up and down. Is it known based on the databases that you have that if I knock down particular genes will resemble my gene list? So you go to this knockdown atlas, and I want to show you something here. So basically what this is going to do, based on its own gene expression, its array studies, it knows that when I knock this gene down, it's going to resemble my list. And this is the BNIP, it's a family member. We're going to see this later on. But again, this is BNIP3-like. Okay? Again, if we knock this out, it makes the genes that it basically looks like the genes that we see in blood cells from sickle cell anemia individuals. And to be honest, I was looking through this with Akshay, and he saw some things that he knows that are basically potential modifiers of this phenotype, and some of these up here. Obviously, if you knock down an anemia gene, it's going to look more like sickle cell. So I guess what I'm trying to tell you with this is that I think this worked, and that what this is suggesting is that this one right here, and we're going to see this with the IPA, I think this might be potentially affected by mutations in this gene. And I'll demonstrate why I believe that. Okay, so this is it for the base space. So basically what we did is we used this program to collect as much RNA gene expression data on sickle cell anemia in blood. From those studies, we basically found a list of about 1,200 genes that are different between sickle cell versus normal. I took those genes and I basically re-uploaded it into this list and found a particular gene here, BNIP3L, or BNIP3L, that when you knock it out seems to represent the genes the changes in gene expression that we see in the sickle cell anemia individuals. Now what I did was I took this list, there's 1,142 genes that are different, and now I've uploaded it into a new bioinformatics program, and that's where we saw some really interesting things there, and I basically want to go over that with you next.